4. What I discussed in my last class, please.
Sir, synapse.
Synapse completed, right?
Yes, sir, it was
Yes,
completed.
sir.
Okay. So almost the nerve tissue is completed. In the nerve tissue, just a few things I am just giving you as a home task. You do it. One is first of all, idea of nerve. Whenever the term comes, the nerve. What is this nerve? Consists of several bundles of nerve fibers and covered by epineuron. Surrounded by epineuron, then you can call it as the nerve, inside which you will give perineurium and endoneurium also. So suppose in cross section is the epineuron, inside which you will get a number of bundles, and each bundle is again covered by perineurium, inside which you see a number of cells are present. Like this. So then here these membranes are called perineurium, then inside you will get it endoneurium. This is the myelin sheath, and the center you will get the axon, the nerve fiber, and here you will get some blood vessels. You will get the blood vessel here. So consider several bundles of nerve fiber. These are several bundles of nerve fibers, and together it is surrounded by epineuron. Each bundle is surrounded by perineurium, inside which you will get the endoneurium, and in the center you will get the axon, surrounded by which you will get the myelin sheath. This is what about the structure of a nerve. Those are layer of connective tissue actually. It's a layer of connective tissue. So everyone, is also a layer of connective tissue. Epineurium, layer of connective tissue. Then perineurium, layer of connective tissue. All are the layer of connective tissue found to surround this. So this is what about the structure of a nerve. So it is the nerve in cross section, transverse section of nerve. If you classify nerve, three types: efferent nerves, efferent nerves, mixed nerves. Nerves are three types: efferent nerves, and mixed nerves. The three types of nerves are found: efferent, and mixed nerve. So you need to remember these things. Efferent nerves made of sensory neurons, efferent nerves made of motor neurons, and efferent and efferent. Actually, both whenever the nerve made of both sensory and motor neuron, called the mixed nerves. I'll give you an example. Suppose,
স্যার
yes.
স্যার ডায়াগ্রামটার মধ্যে মাইলিনেশন কোনটা আর এক্সন কোনটা দুইটা তো সেম প্লেস লাগছে
No, look at this. I am just enlarging this diagram. Suppose this is perineurium. Look. So you see, look one thing. So this is clear up to this. Now this is, suppose each one is here called epineurium. একটু বড় করতে হবে। আচ্ছা, perineurium, then endoneurium, then you will get the, this is, you see, myelin sheath, axon. Is it clear now?
ইয়েস স্যার
So what I say, nerves are of three types: efferent nerves made of sensory neurons, efferent made of motor neurons, and mixed made of both sensory and motor neurons. Is it made of? Is it clear now? Efferent nerve made of sensory neurons, efferent made of motor neurons, and mixed made of both sensory and motor neurons. You know what are sensory neurons? The neurons that carry impulse from receptor to CNS. I'm rubbing it out, this part. Sensory neurons carry impulse from receptor to central nervous system. Motor neuron carry impulse from CNS to effector. Effector means gland, maybe muscle. They are the effectors. So instruction from central nervous system comes to the effector or the muscle or glands through the motor neurons. And the impulse from the receptor, like different types of receptor, we have Pacinian corpuscles, Meissner corpuscles, Pacinian corpuscle pressure receptor, Meissner corpuscle touch receptors. They receive the stimulus and carry it to the central nervous system. So this is the basic thing about the nerves. There are three types: afferent, efferent, and mixed nerves. Already I discussed. Sensory neurons carry impulse from receptor to CNS. Motor neurons carry impulse from CNS to the effector. Now, you can even classify neuron based on the function. Then also sensory and motor neurons are there. So this is what about the neurons. Generally, you get it in the structure of brain. You see, whenever you will see that the any information is required to be carried to the nervous system, that is CNS, for interpretation and analysis. You see, you know that whatever the stimulus it is to stimulate the receptor. Receptor is required to be stimulated to produce an impulse. What is this impulse? Is nothing but the difference in ionic concentration. Then that is action potential. Then it is carried to the CNS. In the CNS, there is a center. You will see there later on. There is to interpret and analyze this, and so that an instruction is made. This instruction comes to the muscle or gland via the motor neurons. So afferent fibers is clear now. Afferent nerves or afferent neuron. Efferent nerves, or you can write efferent neurons. So nerves and neurons, I think the difference is clear. Is it clear to you all?
Yes, sir.
Okay. So this is all. I think I discussed the ependymal cells and also the neurosecretory cells, right? I think I discussed other three components of the nerve tissue: the neuroglia, the ependymal cells, and the neurosecretory cells, right?
স্যার নিউরোগ্লিয়া তো বলনি তো
You know, the supportive cells of the nervous system, the specialized cells of the nervous system which are supportive in function. That's okay, I'm showing you. Can I have it out now? Supportive in function. They do not take part in nerve impulse propagation. This is the neuroglia. There are two types. So this is in short about the neuroglia. Neuroglia actually specialized cells found in the nervous system. They are not taking part in nerve impulse propagation. They are supportive in function. And whenever a nerve cell dies, its place is occupied by the neuroglia. The neuroglia basically whenever star-like neuroglia called the astrocyte provide mechanical support. Then you see that those who are larger neuroglia help in the myelin sheath formation called oligodendrocytes. The macroglia they help in engulfing some bacteria and microorganisms called microglia. So this is what about the neuroglia, considered as the specialized cells of nervous system supportive in function. These are specialized cells of nervous system and are supportive in function. These are specialized as, you can write like this, specialized cells of the nervous system. These cells are supportive in function. Second, third, they do not take part in nerve impulse propagation. Is better. Specialized cells of nervous system, supportive in function, and do not take part in nerve impulse propagation. Clear? This is what about the neuroglia. Any doubt? Clear?
স্যার অ্যাস্ট্রোসাইটের শেপটা কি বললে?
Star shaped. This is the astrocyte. And whenever it is oligodendrocyte, clear? Clear?
Yes, sir.
Okay. Now regarding ependymal cells, can I rub it out?
স্যার একটু
The curve showing the absorption of each wavelength of light by different photosynthetic pigments is called absorption spectrum. Clear? The curve showing the absorption of each wavelength of light by is called absorption spectrum. Simultaneously, if it is action spectrum, the curve showing the rate of photosynthesis is called first of all, try to understand these two. Most important one. First of all, the curve showing the absorption of each wavelength of light by different photosynthetic pigments. Then you can use the term absorption spectrum. But whenever the action spectrum, the different thing. Showing the absorption of each wavelength of light. Clear? Is it clear? Then I am showing you there a curve.
Sir, action টা করে দিন action টা
Huh?
action spectrum টা করে দিন
Curve showing the rate of photosynthesis at different wavelength of light. Photosynthesis occurs at different wavelength of light, and this is the action spectrum that shows the rate of photosynthesis at different wavelength of light. I am showing you with the help of a curve. Can I rub it out? The curve showing the rate of photosynthesis at different wavelength of light is called action spectrum, and the curve showing the rate of photosynthesis, showing the absorption of each wavelength of light by different photosynthetic pigments called absorption spectrum. It shows the rate of photosynthesis, it shows the absorption of pigment, light rays. Hello? হ্যাঁ মধু ও রেজাল্টটা না রেজাল্টটা দেওয়া হয়েছে রেজাল্টটা পাচ্ছি না আচ্ছা দেখতেছে ওকে ফাইল আজকে কাল মধ্যে ফাইল দেবে বুঝছি আমি পারবো না ঠিক আছে রাখে ক্লাস Now I am going to, can I rub it out please?
Yes, sir.
Okay. Okay, see. The wavelength of light. Is the absorption of light by so first of all, chlorophyll A. It is up to this. This is the chlorophyll A. Then chlorophyll B. Then comes to the carotenoids. So this is what about the absorption spectrum. You see, the curve showing the rate of photosynthesis at is it visible or not? Will be visible? Is it visible now?
Yes, sir.
Now regarding action spectrum, So this is the action spectrum. So obviously, if you are going to compare it with the action spectrum, it appears like this. So this is called, you can call it now you can where the yellow, so the rate of fluorescence is more in the blue and red region of the spectrum. It's clear from here. Clear? देखा ना? Is it clear to you? Write down, please. Absorption spectrum. Okay, I'm writing. And chlorophyll B. Absorption spectrum of chlorophyll A and chlorophyll B indicate that these pigments mainly absorb blue and red region of the spectrum. On the other, action spectrum shows that maximum of this takes place in absorbs mainly absorb blue and red type. Blue and red light. That is very easier to you. Now go to this. What is written? Absorption spectrum of chlorophyll A and chlorophyll B. Look one thing. Absorption spectrum of chlorophyll A and chlorophyll B indicate that these pigments mainly absorb blue and red light. Clear? Is it clear? Absorption spectrum of chlorophyll A and chlorophyll B indicate that these pigments mainly absorb blue and red light. Clear?
Yes, sir.
Here, action spectrum shows that maximum photosynthesis takes place in blue and red regions of the spectrum. Look. Whether the curve showing like this, the maximum photosynthesis takes place in the blue and red regions of the spectrum. That is, this one, blue and red regions of the spectrum. Clear?
Yes, sir.
So first thing it is. So I'm going to rub it. It is required to be very much clear actually. It is most important. Can I rub it out? Is it clear? Note it down.
Yes, sir.
Okay. After that, we have to come to most important Emerson effect. Before going to Emerson effect, we should have some photo system also. Okay, before that, we need to go to some two other terms. We have to go through some more terms here. First of all, before going to the Emerson effect, we should have idea about the photosystems also. Photosystem 1 and photosystem 2. And otherwise, we have to discuss the photosystems here first. Then to light emerson effect and also the photoluminescence, photophosphorescence, all are to be discovered. First of the photosystems. In photosynthesis, there are two photosystems participate. Here you have to discuss light and chlorophyll together. Two photosystems participate. Photosystem 1, photosystem 2. Two photosystems are actually participating in the photosynthesis and both are required. In each photosynthesis, there is a reaction center chlorophyll. Reaction center chlorophyll is in photosystem 1, the reaction center chlorophyll is P700 chlorophyll A. In photosystem 2, it is look, two photosystems. In photosystem 1, it is what? P700 chlorophyll A, and in photosystem 2, it is P680 chlorophyll A. What is the basic point of difference here? P700, why the term used as P700 and what is the meaning of the term P680? When the term P700 is used, means it can absorb light up to 700 nanometers.
স্যার P700 মানে
is a chlorophyll molecule having the ability to absorb 700 nanometer wavelength of light up to. Then the term comes P700. P what comes from the photosystem? The chlorophyll molecule of the photosystem that can absorb highest 700 nanometer wavelength of light. There is absorption where absorption peak is 700 nanometer is considered P700 chlorophyll molecule. So P700 actually is a chlorophyll molecule where the absorption peak is found at 700 nanometer. On the other hand, P680 chlorophyll molecule is also a chlorophyll molecule of a photosystem where the absorption peak is at 680 nanometer. Is it clear now? Is it clear? Basically, in each photosystem, a number of photosynthetic molecules are found. Suppose photosynthesis appears like this. So this is photosystem 1. Similarly, photosystem 2, PS2. So they are different pigment molecules. They all are the different pigment molecules found here. So ultimately, the molecule, pigment molecule that takes part in the photosynthesis or in the light phase should be the P700 chlorophyll A, not P680. Clear? Who is to take part? P700 chlorophyll A is to take part in the reaction. So the light wave photons are to be absorbed by these pigments. Remember this thing, although all these accessory pigments, you know, they are the accessory pigments.
স্যার
Hmm.
স্যার
বলো।
ইন ফটোসিস্টেম বি এটা কি P680 ক্লোরোফিল এ না বি হবে?
Hey, B na. B should not be. Both are chlorophyll A. Remember this thing.
So what will happen if light greater than 700 nanometer comes?
Photosynthesis will not take place because they cannot ionize. Actually, one thing I am telling you, the photons, the as I said earlier, the electromagnetic radiation consists of a number of photons. The photons should have the ability to ionize chlorophyll. What does it mean ionize actually? That means whenever this photon hit from the outer orbit of the chlorophyll molecule, electrons required to be ejected out. Are you getting me?
Yes, sir.
If it is not done, then photosynthesis cannot take place. I will be discussing in my next class. You will see that. So that's why the idea of this system is most important. The actually accessory pigments are there. These accessory pigments are have also the ability to absorb light. Actually, although a lot of pigments are there, but accessory pigments perform two roles here. One, Suppose carotenoids may be there in the light harvesting complex. Both are actually light harvesting complex. You can call it light harvesting complex. Both mean there are a lot of photosynthetic molecules are present here, so you can call it light harvesting complex. P680 and P700 propyl are the light harvesting complex. And here what happens whenever photon comes, first of all, the accessory pigments can absorb them and send them to the P700 propyl. One role. Other role is going to play is to protect propyl from photodestruction. The accessory pigments, sometimes questions come. How do carotenoids protect propyl from photodestruction? So what I said, their role is to absorb photons and to send it to the P700 propyl. How I will discussing later on. And P other role is to protect propyl from photodestruction. You know, in photosynthesis, oxygen is produced. I think you know it. In photosynthesis, oxygen is produced, and this oxygen is actually nascent oxygen. Oxygen produced in photosynthesis called nascent oxygen. And nascent oxygen is actually harmful to propyl. It can oxidize propyl molecule. So if propyl molecule gets oxidized means it is actually the photodestruction of propyl. Then what happens? Then obviously photosynthetic efficiency or the productivity decreases. So what these carotenoids are doing? The role of carotenoids is to convert look, carotenoids convert harmful nascent oxygen to harmless oxygen molecule. As carotenoids can convert harmful nascent oxygen to harmless oxygen molecule, we can say, so in this way carotenoids protect propyl from photodestruction. As you remember this thing, when the intensity of light is more, there will be more production of oxygen during light phase. All these are nascent oxygens, so they are required to be converted to oxygen molecules in order to protect propyl from their effect. And that's why the what it is carried out by carotenoids mainly is to convert harmful nascent oxygen into harmless oxygen molecule. That's why such accessory pigments are required to be present in the light harvesting complex. Each one is actually, as I said earlier, light harvesting complex. Is also a light harvesting complex. Is also a light harvesting complex. Here, so only one P700 is there, not that. It contains a number of molecules. Their role, as I said earlier, to protective in function and also to convey the light energy trapped by it to the P700 propyl molecule. Ultimately, from the outer orbit of P700 or P680 propyl molecule only, the electrons are ejected out. Clear?
Sir, are the light harvesting complex also called antenna?
Now, light harvesting complex, the whole. The accessory pigment of the light harvesting complex are called antenna molecules or antenna propyl molecules, right? Getting me? Light harvesting complex includes all. That means you will get even P7. Light harvesting PS1 includes P700 also. But among the except P700, all are called antenna profile. Are you getting me? One is the core. Actually, in the light harvesting complex, there are two parts. One part is called the core. Other is called the antenna part. Core part contains P700 profile A, antenna part contains others. Are you getting me now?
Yes, sir.
Okay. So here what happens? The light energy, whenever it is trapped, it is to be conveyed like this. Now you remember this thing that whether both these photosystems take part simultaneously in photosynthesis, obviously. Both these photosystems are taking part simultaneously. It is not that one is waiting for the other for the adjustment of sunlight. Remember this thing. It is not that one photosystem is to wait for the others for the absorption of light energy. That means both these photosystems can absorb light energy. I am repeating it again. Both these photosystems can absorb light energy simultaneously. Are you getting me? Both these photosystems can absorb light energy simultaneously. One is not going to wait for the other. It is not that P700 is to absorb light energy first. In the meantime, P680 is to wait till the energy it absorbs energy. No, both can absorb light energy simultaneously, but they have to follow different pathway. It is different thing, but they have to absorb light energy simultaneously. Is it clear to you all? Is it clear?
স্যার
বলো।
স্যার লাইটটা যদি 680 থেকে 700 এর মধ্যে হয় তাহলে তো শুধু PS1টা কাজ করবে না।
হ্যাঁ হ্যাঁ বুঝলি। আর বল।
লাইটটা যদি 680 থেকে 700 এর মিডলে হয় তাহলে তো PS1টা শুধু কাজ করবে না।
হ্যাঁ, obviously, but it is not, whenever sunlight comes onto the earth, it's not possible that only one wavelength is coming. Are you getting me? Are
Yes,
you
sir.
getting me?
Yes, sir.
Okay. Now, we have to say another thing today. Light harvesting complex of PS1 and PS2. Actually, you can write there are distinct PS1 and PS2. So it is not that these two are the same. Light harvesting complex are found different, and so you can write in both. One is the core. It is in the center. Antenna molecules is in the periphery. In both these light harvesting complex, one is actually the center, which is actually the core, other in the periphery, which is actually antenna molecules. Core molecules means the reaction center profile molecule. Is it clear up to this? Is it clear up to this?
Yes, sir.
Okay. Okay, I cannot continue now. So actually after that in the next class I have to start from here so that I can complete the live phase, complete the dark phase also. Thank you very much. You will get the result of previous exam by before Wednesday.
স্যার
হ্যাঁ বল
স্যার কোর পার্টটা কি বলেছিলেন?
core molecule actually in the center of the light harvesting complex. The molecule present in the center of the light harvesting complex is called the core. And here basically which one is found? Basically the reaction center profile. If it is a PS1 then you will get the P700 profile A. If it is the PS2 you will get the P680 profile A.
ওকে স্যার।
So I'm going to close now, okay? If you have any problem, you can write to me directly. In the meantime, I'm going to tell you another thing. Okay, next I'll tell you. Now only 25% is there. Okay, thank you very much.